Good afternoon. My name is Stuart Moss, and I'm from the NICHD. And it's my pleasure today to introduce, as the WALS speaker, Dr. Marisa Bartolomei, who is a professor in the Department of Cell and Developmental Biology at the University of Pennsylvania School of Medicine. Dr. Bartolomei received her PhD from Johns Hopkins University and then did a postdoc with Dr. Shirley Tillman at Princeton, where she first started working on imprinted genes, specifically the H19 locus. She has continued her study of imprinting and epigenetics ever since. Over the years, Marisa has made a number of very important contributions to the field. Her lab determined that imprinted genes are regulated by differentially methylated control regions and identified both cis-acting factors and at imprinted loci and also trans-acting factors that are important for setting the DNA methylation program in the germline and maintaining it after fertilization. At Penn, she became interested in the role that the environment may play in altering DNA methylation events of genes and how these perturbations may influence disease. She showed that various procedures used in assisted reproductive technologies, or ART, result in imprinting changes and DNA methylation alterations. Because there are imprinting disorders due to epigenetic dysregulation, such as Beckwith-Wiedemann syndrome, it is possible that these DNA procedures might be detrimental to the developing embryo. More recently, Marisa has turned her attention to another facet of the environment, the effect of exposure to endocrine disrupting agents such as BPA. Her lab has shown that this exposure is associated with increased loss of imprinting and may affect metabolic health across multiple generations in the mouse through stable inheritance of DNA methylation at the imprinted locus, IGF2. And this represents another example of transgenerational epigenetic inheritance that results in developmental defects. In addition to her stellar science, Marisa has always been a great scientific citizen. She has served on numerous editorial boards, and in addition to serving on many NIH review panels over the years, she is presently a member of the NIH Council of Council's Common Fund Evaluation Working Group and on the advisory groups for epigenomics and 4D nucleome. The title of her talk today is Why Parents Matter Epigenetically, Genomic Imprinting in Health and Disease. And please join me in welcoming Marisa today.
Okay, this is on now? Okay. First, I would like to thank you all for heading out in the cold weather to come here. And I would especially like to thank Stuart and the NIH for inviting me to give this lecture. I've had a great day seeing a lot of friends, and I'm hoping that you'll be able to get a lot out of this lecture and be more appreciative of why your parents really do matter, or at least you can go home and tell your children. So let's see if we can get it. So let me just start. What I would like to do today is just give you a little bit of background about imprinting, and then tell you a little bit about our mouse models that we've developed to study human imprinting disorders, but then tell you two stories. One about some single-cell imaging that we've developed that we're incredibly excited about in collaboration with some great new colleagues at Penn, and then also tell you a little bit about some of our studies on assisted reproductive technologies. So this first slide here just gives you the definition of genomic imprinting, which is the unequal expression of the two parental alleles of a gene. So for mammals who have two sets of chromosomes, one they inherit from their mother and one that they inherit from their father, most of these genes are expressed from both alleles, barring some sort of mutation. But for imprinted genes, these genes are either expressed from the paternal allele or from the maternal allele, with the opposite allele being silenced. And so we say that these genes are imprinted. We know that there are about a little bit over 100 imprinted genes that have been described in mouse, and many of which are conserved in human. And so what we've been able to do, because of the conservation between mouse and human, we've been able to go back and forth in studying them in between mouse and human. So what happens, the consequence of imprinted genes, though, the main consequence of imprinted genes is in mammals, you require both a contribution from your mother and a contribution from your father in order to be viable. In other organisms, you have parthenogenesis. But in mammals, you require both maternal and paternal contributions. So what are these genes that are imprinted? When we first identified imprinted genes over 20 years ago, we thought that these genes were mostly involved in growth, that they were important for fetal growth. But now we know that these genes have lots of roles in development. So in addition to either enhancing or suppressing growth, they're involved in memory, in cognition, in behavior, postnatal energy homeostasis, and a whole variety of processes. So these genes do have very central roles in development. What we also know is these genes then are monoallelically expressed, and we know that too much is bad. So if you have a loss of imprinting scenario where you have both alleles being expressed, that's bad. And we know that if you just have a single mutation or some type of other effect where you don't have any expression at all, that's bad. So here are some examples shown on this slide. So I have some mouse mutation examples of the importance of these imprinted genes as well as some human syndromes. So on the left is an example of an imprinted gene, a really interesting imprinted gene called GRAB10, which is involved in growth. And it's a really unusual imprinted gene in that it starts out being expressed from the maternal allele in the somatic tissues, but then later on in development, in neuronal lineages, it actually flips to the paternal allele. This is a really unusual imprinted gene. But when this gene is knocked out or deleted in mice and is transmitted by the mother so that the mother's allele is deleted, but the father's allele, which is silent, is intact, you see overgrowth of this mouse fetus relative to its wild-type littermate. Here's another example of a gene called CDKN1C, which is a growth regulatory gene involved in a number of disorders, including Beckwith-Wiedemann syndrome, which I'll describe in a second. But in this case, deletion of CDKN1C causes placental pathology, so aberrant morphology in placentas upon deletion. So that's mouse, and there's lots of examples. So what about humans? Well, there are a number of imprinting syndromes that have been described through the years, a few of which are shown up here. So Beckwith-Wiedemann syndrome, and these are all rare syndromes, anywhere from 1 in 8,000 to 1 in 20,000 live-born births. But here's an example, Beckwith-Wiedemann syndrome, which is an overgrowth syndrome. And there are two neurobehavioral syndromes, Prader-Willi syndrome and Angelman syndrome, which are very severe syndromes and involve imprinted genes either being deleted or their imprinted expression being aberrantly controlled. So what do we know about the regulation of imprinted genes? And that's really what my lab is, and numerous other labs, including Carl Pfeiffer here at NIH, are interested in really the mechanism of this imprinting control. So what we know is that these genes are actually found in clusters through the genome. So there may only be fewer than 200 in mouse, but most of these imprinted genes are clustered in 1 to 2 megabase clusters, where you have maternally expressed genes, paternally expressed genes. And we also know that for these clusters, they are controlled by a discrete element, element that may just be a couple hundred bases or up to a couple kb, that are differentially DNA methylated. And so DNA methylation, which is, in this case I've shown, is the addition of a methyl group to the cytosine and CG dinucleotides. This differential methylation for these clusters is critical for the proper regulation. So if this element is deleted, you see loss of imprinting and loss of proper expression, or if there's some other perturbation in the system that leads to changes in the DNA methylation state of this discrete element, you see aberrant expression of these imprinted genes. So this element is very key to this possibly 2 kb element is really key to the expression of a set of genes possibly over 2 megabases in size. So just a little bit more background, there are a couple different ways that have been described that these imprinted gene clusters are regulated, and I just want to present one way that's actually used more frequently before I go more specifically into H19 and IGF2. So this is the long non-coding RNA mechanism of imprinted gene regulation, and it really was one of the first examples of how long non-coding RNAs could themselves be really critical for genome regulation. This particular cluster was originally identified by Denise Barlow, and IGF2R, which is expressed from the maternal allele, was one of the first 3 genes that were described as imprinted. So how does the regulation at this locus work? You have IGF2 is expressed from the maternal allele, as well as these solute transporter genes. And then what we have on the paternal allele is a long non-coding RNA that's called the ARN non-coding RNA, and it's over 100 kilobases in length. And the key to the imprinted regulation of this locus is the expression of this gene, which is regulated by this imprinting control region. So the promoter of ARN resides within this couple kb imprinting control element, and when it's unmethylated, ARN is expressed, and it represses IGF2R and these other genes in cis, and likely through transcriptional interference here, and possibly through recruitment of epigenetic modifiers, repressive proteins that repress these genes in cis. On the maternal allele, this particular element is methylated. It becomes methylated during oocyte maturation. That methylation is sufficient to turn off the expression of this gene. That enables these genes, IGF2R and the solute transporter genes, to be expressed on that locus. So it's all about this methylation and expression of this gene. So of the dozen or so imprinted clusters that have been well studied in the mouse and human, this is the most common mechanism for regulating imprinted gene clusters. One that's used much less frequently, but is actually evolutionarily the most ancient, because remnants of it can be found in placental mammals, is the H19 IGF2 locus. So IGF2 is the insulin-like growth factor 2 gene, and deletion of IGF2 results in undergrowth but viability. So mice that have no IGF2 are viable, but they're small. Okay? H19, in contrast, is actually a non-coding RNA. It makes about a 2.2, 2.3 kilobase longer non-coding RNA, and out of the first exon, it encodes a microRNA. And it's still a question about whether the long non-coding RNA or the microRNA has a function. But I think that when this was first studied in Shirley Tillman's lab many years ago, the gene was deleted and there was no obvious phenotype, so we were all sort of sad that maybe this gene doesn't have a function. But I think it's very clear now from work that's being done in a series of different labs that this non-coding RNA does have important functions in development and beyond, but they're very subtle and difficult to detect unless you're very careful in a mouse model. Okay, so how does imprinting at this locus work? Well, the imprinting control region, which is differentially methylated, also serves as an insulator. And so it's a CTCF-dependent insulator. So H19 is expressed from the maternal allele, IGF2 from the paternal allele. They share enhancers such that the enhancer drives the expression of H19 maternally and IGF2 paternally. And what's key to the regulation is this CTCF-dependent insulator. So when it's unmethylated, there are 4 CTCF binding sites in the mouse. CTCF binds, prevents IGF2 from accessing these shared enhancer, and that drives the expression of H19. In contrast, on the paternal allele, this element is methylated in the male germline during gametogenesis, during spermatogenesis. And this methylation is sufficient to shut off H19, freeing up the enhancers to drive the expression of IGF2. And so this this model has been developed through years by the contributions of the Tillman lab and Carl Pfeiffer's lab, my lab, and many other labs. So it's been a lot of work that's led to this. And what we certainly know is that it actually doesn't take very much to distort the balance of expression, change the methylation state, and resulting in aberrant expression of these imprinted genes. So, what I just want to describe to you, we've now actually spent a lot of the last 20 years in my lab building mouse models to actually study this imprinted gene regulation. And never really entirely intentionally, we actually happened upon mouse models that actually mimicked human imprinting syndromes, one of these being Silver-Russell, but more so Beckwith-Wiedemann, the overgrowth syndrome. But I'm actually not going to talk about work that we've done in Beckwith's today. I want to really confine it to this model because I want to use this model to show some very exciting single-cell imaging that we've now been doing in our lab, allele-specific imaging. So this mouse model, as I'll describe to you in a second, is a good model for Silver-Russell. So what was the goal in the original experiment? Originally, we were trying to understand what it took to repress H19 on the paternal allele. And so we knew that there were a series of CpG dinucleotides that were the sites of the methylation, and that was associated with the repression. And so what we decided to do was just start to mutate some of these CpG dinucleotides to see what was sort of the critical point that was required for repressing that paternal allele. And so we had 60 CpGs to work with, and we said, well, which one should we start with? And we decided to start with the ones that were most conserved across different strains of mice and species. And it turned out to be these CpGs that were actually in the CTCF binding sites. This is admittedly started before we really knew CTCF bound to those sites when unmethylated. So we made these mutations, 9 CpG mutations, and what happened was that these CpG mutations allowed CTCF to bind to the sequences. They were not important contact sites for CTCF, but if they were methylated, they blocked CTCF from interacting in those sequences. So when we mutated them and transmitted this paternally, where H19 should be off and IGF2 should be on, what we saw was that CTCF bound, H19 was expressed, and IGF2 was repressed, and we ended up with small mice. And these mice looked pretty similar to mice that were deleted for IGF2. And as I mentioned to you, it turned out that these mice were a pretty good model for Silver-Russell syndrome, because about a year after we published that original story, these papers came out. Silver-Russell syndrome, or Russell-Silver syndrome, it depends what side of the ocean, how you call it, is a small for gestational age. So these children are born greater than 3 standard deviations below mean birth weight for their age, and they have failure to thrive and asymmetries and other types of characteristics. And it turned out that the group that had been studying this had been studying a genetic linkage, but then they started to think actually about the small mice and the imprinting, and they decided to look at H19 and IGF2 in their patients, and they actually found that, that many of the patients had loss of methylation, DNA methylation of this region, with no evident genetic lesions that they've been able to find. So there's loss of methylation leading to biallelic H19 and loss of IGF2. So that actually was very helpful in trying to track that. And since then, it's shown that a vast proportion of the patients with Silver Russell actually have this methylation lesion. Okay, so sort of back to our mouse model. So when we generated this model, we knew that we had biallelic expression of H19, but we also had a small amount of IGF2 present, still residual IGF2 present. And so it was really unclear to us where the IGF2 was coming from and why there wasn't completely 50-50 H19 expression, equal expression for the maternal allele and the paternal allele. And so we started to think about it. So what does this really mean? What does biallelic expression look like at the single cell locus? Is the wild type expression only the maternal of H19, maternal allele of H19 is expressed? But what about, and the mutant we see biallelic expression, but is it because the maternal and the paternal allele is expressed in every cell? Or is it because you have a mixed populations of wild type looking cells and mutant cells, or some combination of that? And why we care about that is because it has really important implications for the mechanism of what's going on. And it also is that we're looking in complex populations of cells, and there may be that certain cell types within the liver or the muscle are expressing the gene wild type, and other types are expressing it in a mutant manner, and that may tell us something about tissue-specific control of imprinting and what the phenotypes in our mice or in the human patients look like. So what we really like to do is look at large populations of cells at one time, but single cells. And so we tried for a number of years to get allele-specific in situ hybridization so that we could look at H19 maternal and paternal alleles in cells, but that really didn't work for us. And so along comes some really wonderful colleagues that joined the engineering department at Penn, and every place needs an engineering department because they have great technology and they're awesome collaborators, and they want to bring their technology to your biological questions. And so Arjun Raj is a new assistant professor, and he has these two wonderful students. Marshall was the first one to sort of come up with some of the idea. And they said, yeah, we're interested in monoallelic expression. We're up for the challenge of doing the imaging on a single cell basis. And so the idea here is if you have two genetically distinct, so DNA polymorphisms, RNA polymorphisms, two genetically distinct strains of mice that have polymorphisms in a cell, can you determine whether the RNA that you detect comes from either the maternal allele or the paternal allele, just based on these polymorphisms? So the polymorphisms, there's just a few, they're over a large region, and by using short oligos, there's not enough specificity to do this. So what Marshall said, well, let's try a different approach, and it's really called a toehold approach. And so what he said was, here's a polymorphism. It's single-stranded, on the single-stranded, but let's take the rest of the oligo and take another piece of DNA and create a double-stranded. So you have a single-stranded part that has the polymorphism and the double-stranded part with no polymorphism. This single-stranded part will come and bind to your RNA where you have that same polymorphism, okay? If it were just that piece, it would flop back on and off. But the fact that you have this double-stranded piece here, once that piece of the oligo binds to your RNA, then what happens is the kinetics of binding is such that this is going to melt off, and this probe will then go on and bind to the rest of your RNA, and it has a floor, so it has a floor that's distinct. And so we have floors for the black 6 strain, the maternal allele, and then we have a different probe with a different floor for the other allele, the polymorphic one. And then the same thing goes here. So what does the experiment look like? So it's sort of a drawing. Here's a fibroblast. And we first take guide probes, and so this has one floor. It will bind to every H19 RNA in the tube. And so it will bind to in the cell. And then we add our maternal SNP probe, which has a different floor, and a paternal SNP. And we look for where the guide probe cross-hybridizes with either the paternal probe or the maternal probe, but never both. Okay? And so only in cases where there's hybridization of one or the other and the guide do we count it. And what does this look like? This is all done by computer. They do the imaging by fluorescence, but the computer then goes and makes all the calls. So here's a fibroblast again. Here's the hybridization. Here's a part of the cell. Here's the guide probe channel. Here's the one probe, the Castaneous SNP probe channel, and the black 6 SNP probe channel. And what the computer then does and makes calls for the guide probe, the Cas probe, the black 6 probe, and then it overlaps them and says, okay, there's these many that are Cas, C7, there's these many that are black 6, and it counts. And it goes through the cells, and it does it automated by Paul's great programming software. So here's our test case. We do this cross, F1 hybrid, Castaneous and black 6, and we developed mouse embryonic fibroblasts from these, and we look at them. So you're going to see a few of these figures. So each one of these lines is a cell that's been quantified. And you can see that the orange correspond to the C7, the Castaneous, and the blue correspond to the black 6. So this is a wild-type cell, which is F1 hybrid, and the Castaneous is being expressed. We know this from qPCR. And what we can see is every cell that we count, we see almost only all Castaneous RNA. But we do see a little bit of blue corresponding to the allele that should be off, because we know this is wild type and it's off. So we then do a control, Castaneous by Castaneous, so there's no black 6. We see about the same amount of black 6 probes. So that's our background. So that's cross-hybridization. Okay, so what does this mutant look like? And this mutant looks like this. These are, again, our cells, and if you do a little outtake, what you can see is that there's two populations of cells. Cells that express both the maternal and the paternal allele of H19 absolutely biallelically, and cells that are largely monoallelic or wild type. So what does that mean? It means that what we're seeing is a mosaic mixture of cells that look wild type and cells that are completely mutant and biallelic. Okay, so because we know that some cells express H19 monoallelically and some express it biallelically, what does IGF2 look like in these single cells? So remember, in the wild-type conditions, you should only see maternal H19 and paternal IGF2, but we know that we see cells that have maternal H19, cells that have paternal H19 off, like the wild type, and cells that have paternal H19 on. So either those cells would have IGF2 on, or those cells would have IGF2s on. So what's the answer? And so the answer is only when you have monoallelic H19, and there's lots of examples of that, do we ever see IGF2. The biallelic cells never express IGF2. And so what that means is these enhancers can either engage H19 or IGF2 on a single chromosome, but they can't engage the two of them at the same time. And so that's really important information for understanding how this locus works. Okay, here's another way I can demonstrate this. This is actually, occasionally we can see the sites of transcription in the nucleus. So here's the nucleus, and here's the cytoplasm. And this is an example of a monoallelic cell. Remember, there's 2 chromosomes. And we see, here's the guide probe. So we see one site of transcription of H19. We know that it's castaneous, but we don't see any of the black 6, so it's a monoallelic cell. And it's in this cell that we see IGF2 expression. If we have a biallelic cell that has 2 sites of transcription, this guy right here is castaneous, this guy right here is black 6, we see no IGF2. So this really convincingly shows us that you only see IGF2 when you don't have H19 being expressed from that particular chromosome. Okay, so in this population of cells from our mutant, we had 25% of the cells actually still expressed H19 monoallelically. We don't really understand why. 25 are still fine. 75 express it biallelically. Those cells have more biallelic. Those cells have more total H19 than the monoallelic cells. But the question is then, what is the difference between that cell and those set of cells and that set of cells? And one of the things that's sort of intriguing to us is in this mutant population of cells, we've did DNA methylation analysis by sulfite clone and seq, and 25% of the strands look like wild type, fully methylated. So each of these is a strand of DNA, and if it's filled, it means it's methylated. If it's empty, it's unmethylated. And so wild type shows you all methylated. And so 25% of these look like this. So the next questions that we wanted to ask was really 2 questions. How stable is that allelic expression pattern? So once a cell becomes monoallelic or biallelic, does it stay? And what is the mechanism for this allelic pattern? So to address this question, we had developed sort of another experiment. And what we decided to do, these are primary mouse embryonic fibroblasts that we are looking at. And they only have a few cell divisions that in vitro, but we plated them out very dilutely on a layer of human fibroblasts. And we let the colonies grow up. And then we imaged them so we could see colonies of cells. And what we saw was, we then asked the question, do all of the cells in a given colony, they are all correspond to the same starting cell, so they're all sort of offspring of that same cell. What does their expression pattern look like when you image? And what it looks like is colonies are either all biallelic or they're all monoallelic. So we see that once a decision is made, it's maintained and it's stable. So now that we could get these colonies, we grew them. We tried to grow them up to image them and get enough cells that we could do the bisulfite on them, and we finally were able to do the imaging, the fixing the imaging and the bisulfite. And what we saw was, as we expected, when a cell looked wild type or monoallelic, it had a normal methylation pattern. And when it was abnormal or biallelic, it had a loss of methylation. So consistent with what we thought was that the biallelic cells showed that the ICR had a loss of methylation. Okay, and so we've now been extending this SNP-FISH into tissues, which is really what we really want to do, because we want to look at complex populations of cells in mouse tissues, or my fellow would like to look in tissues in some of her Beckwith's patients. We can see that there are clonal examples. This is actually cardiac tissue, which was easier to do the imaging on. And you can see that there's patches of cells that either are biallelic or monoallelic, suggesting, again, that you have a cell that goes through some sort of decision process, either loses all its methylation or it maintains its methylation, and then that's stable. Okay, so let me just summarize. What I've shown you is we now can image allele-specifically using polymorphisms at the single cell level, and we can do it both in cells and in tissues. What I forgot to tell you is that when you quantify your signals you get from the microscopic imaging, the SNP-FISH, it actually is essentially identical to what you see when you do bulk cultures by RT-PCR. And that we end up with an epigenetic mosaic mixture in these loss of imprinting mutants which is stable. So what do we think has gone on in our mouse mutant? So we know that with that particular mutation, when it comes from the father, it has methylation of the CPGs that are remaining that could be methylated. And that methylation is sort of present a little bit in the blastocyst, but it begins to be lost. And what we think is, and this is a period where there's a lot of reprogramming that goes on in mammalian development, but once you get post-implantation, the reprogramming is done in the somatic tissues, and the maintenance methyltransferase is then reinduced and is in place. And so what we think happens is every cell has either made a decision to lose its methylation and be a biallelic, or maintain its methylation and be monoallelic, and then that's stably maintained. And so the monoallelics look wild type, and the biallelics have this loss of methylation. And you can treat it with DNA methylation inhibitors and actually knock a wild type looking cell into a mutant looking cell. Okay. So that sort of is the imaging, and we would like to now use that in a bunch of other systems. But with just in my remaining few minutes, I just want to tell you about another project that we've been working on. This is a collaboration that I've been doing for many years with Richard Schultz at Penn. And this is, Stuart mentioned that a little bit in his introduction, our work on assisted reproductive technologies. So it's been known for a long time in the large animal world, where they often use in vitro fertilization to propagate strains, that these embryos, once they're transferred back to moms, can end up with large offspring syndrome, which ends up with a number of problems, and placental and other types of problems. And we also know that some of these things have been observed in the mouse. But what had been a little bit less appreciated until more recently is that there is some increase of problems, and still low, but some increase in problems that are associated with assisted reproduction. And we know that there's preterm labor, preterm birth, low birth weight, some increase in congenital disorders. There are other problems that are very controversial in the literature about whether there's autism or other things like that. And I think there's a really important study that was done by an ICHD that just came out that sort of suggested that there wasn't an increase of autism in ART, which is, I think, important. But what we've been interested in was the observation that there seems to be an increase in imprinting disorders, which, remember, are still very rare. They're still, about 1 in 10,000, so you need incredibly large cohorts to really see an increase. And some of the studies that have been done, for instance, with Beckwith-Wiedemann, suggest there's a 6-fold relative risk in seeing Beckwiths in assisted reproduction children. So when I talk about, let's just go a little bit over assisted reproduction, particularly in vitro fertilization. So what happens here, you have egg production that's stimulated by hormone therapy, and the eggs are retrieved. They're then combined with sperm in a petri dish, allowed to fertilize, or in some cases, the sperm are injected into the eggs in the case of ICSI. They then will grow for a couple division in the dish and then transferred back to mom. So you have superovulation, you have embryo culture, and you have embryo transfer. And what's really important about this is these particular procedures take place at a really sensitive time in development when the genomes are being reprogrammed. So, for example, shown here this is DNA methylation, increasing DNA methylation sort of genome-wide. And you can see that PGCs, you lose DNA methylation, there's reprogramming, and then it's reacquired. And then after fertilization, it's lost again as the genome is being reprogrammed and then reacquired. And superovulation takes place when oocytes are reacquiring the DNA methylation during maturation. Embryo culture and embryo transfer takes place when the genome is being reprogrammed, going from specialized eggs and sperm into pluripotent cells, and then determined into cells that are going in various lineages. So you see all this DNA methylation reprogramming. Importantly, imprinted genes after fertilization have to maintain their identity, have to maintain that DNA methylation. So when the genome is losing a lot of its DNA methylation, imprinted genes maintain it by mechanisms that we don't really understand fully at this time. So we and others have done lots of experiments using the mouse model to address these questions, ultimately why there are an increased risk of epigenetic perturbations and imprinting disorders in ART, children conceived of ART, with the ultimate goal of making it safer and trying to understand which procedures are contributing to this. So we and others showed that there was a loss of imprinting and loss in DNA methylation in the mouse and imprinting control regions after embryo culture. There was loss of methylation that's associated with superovulation. A former fellow in my lab, Rocio Rivera, showed that there was loss of imprinting, loss of methylation that just could be associated with embryo transfer. Take an embryo out of one mouse, take it out, put it into another mouse. So an environmental perturbation. And we recently showed that you had more severe loss of imprinting and loss of methylation when the entire IVF protocol was used. But that was really focused in the placenta where you saw the greatest changes. So the questions, that most recent question that we asked with two fellows in my lab, Eric DeWaal and Lisa Vrooman, was, can we associate these specific procedures that used in assisted reproduction or IVF, in vitro fertilization, with these various pathologies that we observed? And specifically, I'm going to focus here on the placenta in the last few minutes. So what Lisa and Eric did was they designed an experiment where we could distinguish the various procedures. So we first have natural mating, so mice naturally mate. And then what we're going to do, we let them gestate to term, and then at term we sacrifice the mouse and remove the embryo and placenta. Our more recent design, we cesarean deliver the mice and foster them and take the placentas, and we're trying to associate the placenta with the phenotype of the offspring. But right now I'm just going to talk about term placenta and a little bit on term fetuses. So that's the natural situation. And then we have this control where there's just purely embryo transfer. So the mice, there's no superovulation. The mice mate, we remove the embryos, put it back in, a foster mom, gestate, transfer, and then we sac. And then we have superovulation plus embryo transfer. So we superovulate the mom, let them mate, take the two-cell embryos out, transfer them to a foster mom, gestate, okay? And then finally, the whole IVF procedure of superovulation, culture, IVF, culture, transfer, okay? And so I'll just summarize the findings on the next few slides. So the first thing we looked at is the placenta, and this is placental weight. So what is shown is each of these graphs will be the same with the natural, the embryo transfer, superovulation, embryo transfer, and IVF. And what we see is that either by placental weight or placental to fetal weight, we see that all of these conditions result in an increase in weight relative to the embryo. So just embryo transfer alone. And then you see sort of it worsen when you use the whole procedure. And so here's an example. These are H&E stained placentas. And so you can see, here's a natural placenta, here's the IVF. It's pretty obvious that they're larger. You don't need a lot of degrees to see that. Even I can tell that. And so then what Lisa and Eric did was then they stained and they looked for the junctional versus the labyrinth zone. And this is pseudo-colored here just to show you, here's how much you expect to see of the junctional zone versus the labyrinth. The labyrinth is where you have exchange between mom and fetus, and junctional has some endocrine secreting cells, et cetera, but that's just a very basic difference. And you can see that as you go on, especially once you get to IVF, the junctional zone relative to the labyrinth is greatly expanded. And when you do the measurements, and this is all done blinded, this is like one of the most important experiments to do blinded that I've ever come up with. We actually have a fleet of undergraduates in the lab that we hand these to and have them do look at the area. And so what we see is that natural and embryo transfer don't look statistically different, but once you get to superovulation plus embryo transfer and IVF, you see enhanced junctional to labyrinth ratio. So then we looked at some molecular biology of these placentas, and one of the things we looked at was DNA methylation of these imprinting control regions, and this is done in bulk. You have a methylated allele and an unmethylated allele. Normally, you would see 50% methylation if you're not doing it allele specifically. And what you can see is for these 3 imprinting control regions, there's reduced methylation that's statistically significant only in the IVF. And similarly, when we look at genome-wide methylation by the LUMA assay, we see reduced global DNA methylation. So in this case, only the IVF show methylation changes. I was just going to show you real fast what do the fetuses look like at this point. This is expression, allele-specific expression. And you see a couple fetuses that have some cells that look biallelic. This is really where we'd like to do our allele-specific SNP-FISH so we can see just how many cells are they certain types of cells. But you do see statistically more expression from the normally repressed allele. The allele should be off, so at this basal level, in the IVF versus the natural. But actually, if you look at the ICR methylation, the methylation's not really that bad. So there's probably just a few cells in that population that are expressing the normally repressed allele really highly. But what we did see was that in the case of H19, when we had a mouse that had reduced methylation or tissue that had reduced methylation or biallelic expression, that we would see it in the brain as well as in the liver. So the mistake happened early in development and was maintained. Okay. However, this is placenta, and placenta shows much worse phenotype than what you see in the mouse. And most of the time, the fetus looks fine, but in this case, the placenta is very adversely affected, and adverse placenta is really associated with later problems. So this is just a summary of what I've shown you. And what this tells us is that the more manipulations we introduce, the more severe the placental and in the end, embryonic phenotype. So with just embryo transfer, we only see an increase in placental weight, but when we introduce superovulation, we see the junctional grown overgrowth. And when we introduce all of the procedures in IVF, we see perturbations in DNA methylation, gene expression, and much more serious than we observed everything. So again, this shows us that each of these procedures contributes a little bit, and so each one of them may have some kind of require some kind of adjustment. And we already know now that, for example, in terms of superovulation, women are given much lower doses of hormones when they're being superovulated, which I think is an important thing. And their culture is being optimized, and lots of things are being incorporated. So I think this is a really nice system for studying changes and procedures in assisted reproduction. So what we're moving on to do is to figure out if these morphological changes in the placenta can be associated with aberrant outcomes later on. And we've developed a very nice strategy to C-section these mice and transfer and mark the mice that we can track back the placenta to them and study them. And we're also profiling all the tissues to figure out the extent of the changes. And we really want to know why we see loss of methylation, because we really believe that's really the cause of a lot of the problems that we have observed in these mice. So finally, I just want to acknowledge the people who did the work. This is a nice sunny day at Penn with our former leader, Benjamin Franklin, shown here. The people who contributed to the work that I described today are in purple, together with our collaborators, Arjun Raj and his lab for the SNP-FISH, and then a longstanding collaboration with Richard Schultz and many people in his laboratory, as well as many former members of my own lab. And I probably wouldn't be standing here today if it wasn't for the fact that I had very generous support from the NIH as well as some other organizations. And so I'd really like to thank you for your attention, and I think I can take questions. Okay, thank you. Any questions?
Yeah, when you're talking about the question of why the placenta would be the most affected, it would seem almost obvious, because in one of the first events that happens when you would transfer a developed embryo or zygote to a surrogate uterus would be implantation and separation of the embryo and placenta. Right. Therefore, it's missing whatever internal stimulus, epigenetic stimulus, while it's in process of IVF. You know what I'm saying? I'm saying that, in other words, if in the process of when you're doing artificial, all the things on the outside before you put it in there, it should be having the benefit of being within the maternal environment.
Well, I mean, I think that's an important question. I mean, there's also the question of whether there's genetic differences between mother and offspring, but that's still the case. I mean, there's still an important question of why that happens and if there's a way to remediate the situation, because you can't, in IVF, you have no recourse. You have to transfer it to another mom. It's happening in Petri dish. It's going back into the mom. It's not sitting there the whole time. So I think that's an important question.
Okay, I think that it would be odd if the placenta was not the thing that would be most affected.
So I think that a lot of what we've found when we've been doing a lot of manipulations in endocrine disruptors and everything is any adverse environment seems to affect the placenta more than you would have understood. So I think in some of our endocrine disruptor work, where there's no taking out and putting back in, it's just an exposure, we see similar kinds of placental phenotypes. So there's something else that's going on in addition to what you've brought up. And so I think that the placenta is very, very sensitive to any adverse environmental insult. And that, I think that mechanistically trying to understand what it is when we look across our different systems is really an important question that we're trying to address.
Yeah. Just a point, I had done a fellowship in perinatal neonatal pathology in humans, and I did quite a few autopsies on abortions, and we would always include in the autopsy a placenta and a fetus. In other words, that's part of the basic organ. Mm-hmm. And that's kind of by classic, the first thing we look at in the placenta. That's why it's leading me to the conclusion based on experience also.
Right, but the other question is that why you see increasing procedures give you more adverse pathology too. So that's another question of what we're trying to understand also. So.
All right. Well, thank you very much.
Other questions? I know in a big room, people always are embarrassed to ask questions. Stuart, you can ask a question. Yeah.
What about the next generation?
Yeah, so we haven't done that yet, and I think that's a question that people want to know. Some people have done some experiments where they say, are the germ cells affected? And I would say that in those mice, now they're dead, so I can't do anything with them, that had lower methylation in multiple tissues, you would expect that their germline would potentially have a similar sort of defect. They could have a defect if there's a question of aberrant methylation and remethylation. And so we need to do a larger study, and we're going to first look and see whether, and we're trying to identify phenotypes, certainly one is a placental phenotype, and other phenotypes that we can then look into in the next generation to see if any of them are transferred, yeah. Because the endocrine disruptor work, we now have phenotypes in the third generation, which says that they're transgenerational, so we'd like to see if other environmental insults can be transmitted. I hope not, but.
So your model in the first part sort of predicts that the severity of a Beckwith patient will be all about the timing of the epi mutation, right? And that if you looked at different patients that were more severe, you would expect to see more of these biallylically expressing cells. Is that correct?
Yeah, I mean, I really believe that the more severe ones, it happens earlier, but it's really difficult accessing the tissues. I've been told that there have been some patients that don't look very severe, but they'll end up with a kidney tumor, and by blood they have a low level of effect, and they go in and they take out the kidney tumor, and they take out the adjacent tissue, and they see that there's much worse epigenetic profile that's associated with it. So it's not necessarily easy to detect, but in cases where they've been able to look, that seems to be consistent. And we would suggest that I think that those Beckwith epimutations are maintenance problems, and sometimes it happens earlier and it recovers, sometimes it doesn't. That's why we really are trying to generate a mouse model to be able to study that, and we have so far been unsuccessful. But.
And another question about the second part. So it seems like in some cases the phenotype on allelic expression is more dramatic than the DNA methylation phenotype.
Yeah.
So, I mean, have you looked at, it's much harder to assess, but there's a lot of chromatin marks which could be affected by environment. And how do you start to tease apart that some of those may be really what's responding and it's not so much DNA methylation?
Yeah, so there's never enough time to really talk about that. So I would say, so when you look at the expression, there seems to be all kinds of expression from the repressed allele. And I would argue that it just takes a couple cells to be blasting expression to all of a sudden see what looks really bad. So when you're assaying a big population of cells, you're only assaying the cells that express the gene. But when you're looking at DNA methylation, every single nucleus gives you a signal. And so my prediction is, if I use that SNP-FISH assay, which I love, and I think we're close to that, I would predict where you see those blasting things, but the methylation looks fine. My prediction is that only a few cells are expressing biallelic. But if more are, then we do have to go look at chromatin and other kinds of things. And that would suggest, you're right, that the DNA methylation is not as predictive as you would suggest. I mean, it's an important point. And I didn't tell him to ask that question.
So if there's no more questions, I invite you to accept the exploratory.
Thanks.